Hi, my name is Edward Lammer, and I'm from the Children's Hospital Oakland Research Institute. We do research on congenital heart defects, which are common major malformations affecting between a half and about 1% of all normal newborns. We study both genetic and environmental causes of a particular type of heart defects called conotruncal defects, and this research is supported by the National Heart, Lung, and Blood Institute. To identify genetic causes of these heart defects, we use a variety of experimental human genetics approaches. One of those approaches is array comparative genomic hybridization, which is a technique that detects chromosomal microdeletions or duplications and can detect these abnormalities on a genome-wide search. The second technique we often use is something called MLPA, which is a technique that detects even smaller chromosomal microdeletions, but it's not a genome-wide technique. It's a technique that we apply to a specific group of genes that we think might be contributing to congenital heart defects. These two techniques are useful for detecting genes that can cause congenital heart defects through a mechanism of copy number variation. Copy number variation means that a child is born and has either a lower number of a specific gene than that child should have. For most genes, every person is born with 2 copies of a gene, and some children also are born with 3 copies of a gene, what we call a duplication. Using the MLPA approach, for this project we screened several genes that are known to contribute to normal heart development in mice, especially to the part of the heart that we know is affected in children who are born with conotruncal heart defects. One of the genes that we targeted is called ISL1, and ISL1 is a candidate gene for conotruncal heart defects because of abnormalities of this gene in mice cause similar types of heart defects in the mice. Our study population for this project were children born to women who were residing in 3 California counties and who delivered babies between 1999 and 2004. During that time, the population base was about 1 million births to women residing in those 3 counties. From this population-based sample, we identified every child who was born with a conotruncal heart defect. And among those children, we enrolled and have investigated 389 infants who were born with either tetralogy of Fallot or detransposition of the great arteries. These 2 malformations compromise about 75% of the children who are born with any type of a conotruncal heart defect.
My name is EdKazu, also Egawa. From this study, our MLPA results suggested multiple exons deleted from a Hispanic female with isolated PTGA. Detailed molecular analysis revealed the region of about 20 kb region was completely deleted, but only islet 1, but not involving the other flanking genes. We show that this 20 kb deletion was created by non-homologous endojoining. So the significance of this, our result, are summarized into 2 categories. Perhaps this micro deletion is probably too small to detect by routine clinical microarray testing, and which is widely used clinical microarray needs to be modified to detect such a small deletions of islet 1. Also, this is the first report of a mutation of islet 1 from an infant born with isolated heart malformations.